Uh, Hi everyone, I'm Cátia Moutinho, and I'm the Associate Director of GWCCG. And we are officially starting the series Single Cell Seminar series. And this is an amazing collaboration between GWCCG, 10x Genomics, and Millennium Science. And I will pass the word to Geoff McDermott, the Science and Technology Advisor from 10x and one of the main organizers of these seminars. Hi, Geoff.
Awesome. Thanks, Cátia. And thank you all for coming along today. I really appreciate it. So as Cátia said, this is a monthly seminar series that we're kicking off in collaboration between 10x and GWCCG, in that we want to have basically once a month, a 30-minute format session where we have a whole bunch of goals that we want to deliver to you as a new or experienced single cell user. We really want to showcase assay capabilities across the 10x Genomics product portfolio and really show you what you have access to within the core facility here that Cátia runs. We want to offer glimpses into upcoming product capabilities and launches so you know what is coming from us and when to plan for that and how that might affect your research. We want to deliver data, best practices, and importantly, some tips and tricks from real-world users. So not just me from 10x, but having people who have actually used the product in their hands and what are their experiences like and what are things they've learned. Definitely some guidance for overcoming bioinformatic hurdles. We want to make sure that we've got some sessions in there. We completely understand that can be a challenge for some people when dealing with their single cell data. And really our overall goal of this series is to help break down barriers for you to be able to begin your single cell or spatial transcriptomic experiments. So really we want to, like I said, do that with a series of 30-minute format seminars over the next couple of months. Just quickly before we start today, I just also want to highlight that within 10x Genomics, we've internally kicked off with an initiative we call Meet with a 10 Expert. My email address is in here, so note it down if you would like to participate in this. It's essentially if you've got questions about anything single cell or anything spatial transcriptomics, from experimental design to what are the products, how do they perform, to you're already generating some data and you want to know a little bit more about next steps, perhaps budgeting, grant writing, anything. Feel free to reach out to us, and we have Yvonne and myself, who are science and technology advisors, and our field application scientist, Catherine King, who are more than happy to sit down with you. But for today, let's get started with, funny enough, our talk from Cátia and Nguyen on getting started with 10x Genomics Single Cell Experiments. And I'll just provide a really quick introduction to Cátia and Nguyen. So Cátia is the Associate Director of the Garvan-Weizmann Center for Cellular Genomics. She's leading the wet lab research of the Single Cell Genomics Division team. She's a biologist by training with a PhD in biomedicine and a master's in clinical trials and medical affairs. She has extensive experience in the wet lab with a strong background in single cell technology, as well as cancer genetics and epigenetics. So in very safe hands here with Cátia today. And then Uyen is a molecular scientist of the Garvan-Weizmann Center for Cellular Genomics. She is helping researchers perform their 10x single cell experiments in order to uncover disease mechanisms. She obtained a PhD in medical science and a master's degree in protein chemistry. In addition to her expertise in molecular biology, she has an extensive experience in cell biology, which allows her to precisely examine the cell health before capturing. So again, we've got two real-world, very expertise users here today to tell us all about how they recommend getting started with 10x Genomics single cell experiments. So take it away, guys.
Thank you. Thanks, Geoff. I will share the screen. Yeah. So as Geoff told, today we will talk about sample preparation, and me and Uyen, we will tell you what you have to keep in mind. The outline will be, I will talk about biological samples, sample preparation protocols, and then Uyen will continue, and she will talk about the 10x single cell capture workflow at the GWCCG facility and the importance of a good input, the cell input on these type of experiments. What type of biological samples can we have? And we can do single cell experiments. So we can have the liquid biopsies, blood, CSF, BAL, bone marrow. We can have the solid, that are normal or diseased tissues, like tumors. Organoids also is an option. These tissues, organoids, can be from, for example, mice, or from humans, zebrafish. So all type of organisms that have the polyadenylated mRNAs. In the middle, like kind of irid, we have the adherent cell lines. And here, what are the ways that we can have our samples preserved? Ideally, we should always use fresh samples. Why? Because we need cells that are viable and good amount of cells. So ideally fresh sample. Then if we don't have, the second best option is the cryopreserve. These samples need to be preserved in DMS, 10% of DMSO with FPS, and we put in liquid nitrogen. We can also have snap frozen samples or methanol fixed. In the case we have fresh and cryopreserved, we can extract or isolate cells, total cells, and we can do our experiments with these inputs. In the case of snap frozen methanol fixed, the cells are already dead. So what we can do is isolate nuclei and work with that to do single cell nuclei assays. In all these cases, and I will explain after a little bit more, we can always work with nuclei. And we need always to think a little bit what we will expect from our sample, meaning, will we have a big or low amount of sample material, good quality or bad quality? It will be heterogeneous, a lot of cell types, homogeneous. How it will be the cell viability, the amount of cells. So all these things that I'm like these questions we need to know, because knowing these answers, we will adapt our protocol of sample preparation. And where can we find these protocols, sample preparation protocols? Yeah, we always have to choose the best one for our tissue. And we can have lab SOPs, like labs that you already have a connection, networking, protocols IO, PubMed, bioRxiv, ResearchGate, the support webpage of the Next Genomics. So what we have to go is to search, but even if we find the protocol where they are isolating cells from the same tissue that we will work with or we want to work with, we need to optimize our protocol. Why? Because the same protocol can be interpreted differently by different people. So we really need to optimize. Besides the reagents, they can seem similar, right, from the same brand, but the lot number, for example, can change, and this can lead to like different results. The same protocol, different results. So we really need to optimize everything. And all these protocols, basically depending on type of sample, they want always the same, like that we have always the same goal. In the liquid samples, the protocols, they aim to separate the cells from the debris because our cells are already in suspension. And here we can use to separate our cells, liquid gradient protocols, FICOL and FOPREP. My advice here is to use these type of tubes. Why? Because when we use the gradient, liquid gradient, what we do is very dense, the liquid, we put on top our sample, and there is the formation by centrifugation of different phases. So these phases are very, they are not very separated. If we touch with a tube in some place, or the centrifugation is not well done, they will mix these phases. So these tubes keep the different phases in place, and it's easier for us to isolate the cells. Then in case you are working with blood, if we have a lot of red blood cells, use always a lysis buffer to remove these red blood cells. We can also use with liquid samples, magnetic beads. Make sure that you are using nano beads, okay? Concerning the solid samples, here the aim is to obtain individual cells in solution with high viability. Viability is really important for single cell experiments. The first step is to dissociate our tissue. This can be done, the first is mechanical with blades, scissors. We mince our tissue, then we pass through a falcon. And here we do the second step, that is the enzymatic digestion. We always need to find the ideal cocktail of enzymes. And we need to optimize. Try always to use less than 30 minutes because this will stress less the cells. The cells will be stressed because the enzymes normally they work at 37 degrees. So less amount of time, it will be better for our cells. And also what we do is when we put the enzymes, we put at 37. After 5 minutes, we go and we pipe it with 1 ml. So pipe it not very harsh, but try to pipe it for like help dissociation. And we can also use in some kind of tissues, there are already the cold proteases. These are enzymes that work, instead of working at 37, they work at 4 degrees. So it's better, it's less stress for our cells. Then we have to clean, and this we use filters. And here we always filter and wash with PBS with a little bit of FPS. We start with 100 micrometers and we can go until 40 micrometers. It depends always the size of the cells that you will work with. You need always to have like an idea of if your cells will be very big or small. For example, big, the cardiomyocytes, small, PBMCs. And we always need to do gentle centrifugations. Normally, we don't centrifuge more than 300 Gs, more than 5 minutes, because again, be gentle with your cells. They need to be alive. For adherent cell lines, we start from the enzymatic dissociation. We don't need to do the mechanical one. And if you have a sample with high debris, what you can do, there are some solutions. So for example, in the facility, we have fax sorting you can use. Always for the fax sorting, independently what you want, like to select some type of cells, remove the debris, use always 100 micrometer nozzle. The nozzle is like the needle where the cell needs to pass, and also try to use low pressure. We collect our cells always in tubes that already contain an amount of liquid. Like this, the cell will not just reach an empty like tube. It has some liquid, like if we are jumping to a pool, right? If there is water, it's better. Then we can use also the max. Here, my advice is use only if you have high amount of sample, because high amount of liquid, because if you have small, maybe you will not be able to like fill all the column and you will lose your sample. You can also use other option besides these two, the laminar wash. If you have, okay, you remove your debris and you have a solution of cells that the cells present low viability, if your sample is really precious, you can do a cell dead removal. If you have more sample, just repeat, try to optimize your protocol. But if you don't have any other option, you can use, when you have high amount of cells, high volume, you can use a dead cell removal gradient. It's the same as before, FICAL, lymphoprep, you can do that. You can use magnetic beads also, fax sorting. In the case that you have low amount of cells and low volume, what we are using in our lab is the Annexin 5 removal kit. This works very well. If you still have low viability on your sample, you have to think if it's worth it to do nuclei isolation. So nuclei isolation normally are ideal for snap frozen tissues, tissue that is put right away in the liquid nitrogen, so the cells are dead. Postmortem material, or tissues that are very difficult to dissociate, like brain tissue that you have, we have the neurons, so it's very difficult to separate the cells. Here, for the nuclear extraction, you always need to check the buffer that you are doing nuclear extraction, because it needs to be compatible with downstream techniques. It's not the same to do a nuclear extraction for 3' or single cell RNA-seq than for the multiome kit. You need to be careful here. And you also need to optimize the extraction time, meaning you have to do a time course and see where you find like nuclei isolated and some cells. This means that your nuclei were not completely like the membrane is still intact. Okay, the lysis it was not very like harsh. And I will now pass to Uyen. Yeah, she will continue.
Thank you, Cátia, for your fantastic talk and your useful information about the cell preparation. I share this. Yeah, so my name is, so today I would like to talk more about how you can submit your sample to the facility for the 10x single cell capture. So if you are having any single cells planning to do with us, please send us an email and subject your project form. Then we will discuss your project and get back to you with some important tips to start your experiment. So after that, the customer will prep the samples and they will decide whether they want to sort the cell to enrich their target population, or they want to improve the sample quality using the dead cell removal, or if they want to use the cell surface protein staining or the multiplexing samples in a pool. After that, the customer will handle the sample to the facility, and the facility will check the cell viability and the total cell count. So if the cell viability is higher than 70% and we have sufficient cell count, then we will proceed as requested. If the viability is less than 70%, if the cell viability is higher than 70%, but we have fewer than cells as expected, they will still proceed the best effort to capture as many cells as possible. So if the cell viability is less than 70%, we will discuss with the customer whether they want to proceed on their own risk or stop the capture. So the input cell quality is very important for the 10x single cell capture and for the downstream analysis. So what are the criteria we need to achieve before proceeding to the 10x single cell capture? So first of all, try to minimize the debris in your sample by regularly filter your sample with 70 micron and or 40 micron. Cell viability, try to achieve a higher than 70% cell viability, and with optimal cell concentration between 700 to 1,200 cells per microliter for the accurate counting. A recommended recovery cells between 500 to 10,000 cells. And you need to optimize your resuspension buffer to see which buffer give the best viability for your samples. For example, you can use one-time PPS, 0.04% PSA, or you can use your cell culture meter with up to 10% of FCS. If you need any more information, then there are a lot of resources from support.10xgenomics.com, or you can contact GWCCG for the summary of the cell preparation guideline. So in details, I would like to give some example of gray and pool cell suspension. The first example is the panel A, where you can see this is the gray cell suspension from two different samples. So most of the samples, most of the cells are separated nicely, have a ground shape, shiny, and a little bit of dead cells, and the cell viability is about 90% viability. So opposite to the panel A, in the panel B, you can see those two different samples have a lot of debris. And the first image in the panel B gives you an example of the debris actually come from the FCS, the human serum that you use to make up your resuspension buffer. To the key point is please filter your FCS before use it to make up the resuspension buffer. And the second image shows the debris actually come from the human tissues during the single cell isolation protocol. The panel B give an example of gray nuclei suspension. As you can see that those two different samples have a gray suspension of nuclei because they have no debris, no clump, and the nuclei membrane is still intact. Opposite to the panel C, in the panel D, most of the nuclei has the compromised membrane, and it appears as the ruffled and show the evidence of the flapping. So flapping is the loss of nuclear membrane coherence with the nucleoplasm. So it's normally caused by over-lysis. So the key message is always optimize your single cell isolation protocol and the lysis protocol in a nuclei work in order to have a successful single cell capture. So on behalf of the GWCCG, I would like to say thank you very much for your attention, especially to the customer, internal and external, for your support. So we actually learned a lot from the researcher, and those experience actually help us to improve the quality of the service in the facility. Especially thank you to the 10X Genomics and Millennium Science for their fantastic support and excellent customer service. I would like to end my talk today, and I'm interested to hear some more questions and your thought about the talk. Thank you.
Thanks, Uyen.
Awesome. Thank you so much, guys. That was very insightful, and again, a good way to open up the seminar series with understanding what is needed before you actually start your experiment. Your sample prep works, and sample prep, you know, that's where your experiment honestly is going to be successful or it's going to fail, is really at the sample prep step before you even do any of the single cell workflow. So these are really good insights. Does anybody have any questions for any of the panel here today?
I see.
If there aren't any questions, we can definitely give people some time back. Oh, there is a question.
Okay. Yeah. Can you please clarify filtering using 170 or 40 micrometers? Yeah. So I can start with my experience. Uyen, you can then say what you think. Basically, it's like when you are dissociating the tissue, the first filter should be 100 micrometers, just to remove all the clamps, all the big pieces of tissue that were not digested. But then we always filter after, like it's a serial filtration, 170, and only in some cases we filter with 40. But this is when we take our sample to Uyen, and she said, yeah, you need to filter with 40. So she decides, basically. Yeah, it depends a lot of the debris and the tissue. Uyen, what do you think?
Yes. So actually, in my experience, even if the sample very clean, but if we still filter the sample through the 40 micron, it's actually help a lot for the assay's performance, because I have the feeling like in the sample resuspension, we may have some kind of mucus or sticky thing in the solution. We couldn't see it, but as we filter through the 40 microns, so it's reduced the chance of the failure. So I found that if we have plenty of the sample, and if we don't worry about the cell size, then I would suggest filter the sample through the 40 micron to increase the chance of the successful capture.
Okay. Sounds good.
And also because the microfluidic chips in the 10X, the size only about 50 to 60 micron. So if we can do the final filtering with 40 micron, then that would be fantastic. Yeah.
And what type of filters do you use? Like, we use ones that we put on the Eppendorf, and they fit very well. But you use ones that are very small, right?
No, I use that one. I use the one it can fit into the Eppendorf. Yeah.
Okay. Yeah. That's good. Yeah.
Do you ever find any challenges of filtering the sample through those little FlowMe tips that fit on the Eppendorf? Is there a particular sort of cell concentration that needs to be used with that, or any fragile cells that you sort of recommend avoiding it?
Yeah, I'm a bit hesitant to use the FlowMe Trainer because I feel like I don't know, because I just feel like the spore in the filter may be very small, or sometimes we need to brush the sample through the filter, and it's maybe not good for the sample. So I just not feel confident to use the FlowMe. Yeah.
Yeah, I have also the same experience. I rather prefer to use the others, because even if you really don't have a lot of sample, what we do always is to pass first 1 ml of PBS to wet the filter, and then our sample. With others, you cannot really do that.
Yeah.
So yeah. So there is other question. If a researcher has samples that is acquired late and require a lot of prep time, is there options for the facility for late capture, or are there reference recommendations for storing sample till next day for capture? This is difficult. So
yeah, so I think it will depend. So most of the time, the sample they don't have the option to store the sample for the next day. So normally at the moment, the facility still offers some exceptional cases. So if it's happened, then one of the member in the team will have the researcher to capture the samples. Yeah, but normally if the researcher can have the sample, can organize the samples, then we would discuss and ask the researcher to prep the sample as early as they can. So then the capture won't happen at the late time on the day.
Yeah.
And I will say that, within 10X as a company, we definitely recognize that as a limitation for some people, that you might not, samples come from operating rooms. They come from all sorts of places at all sorts of times. And, we hear the feedback quite a lot of, it would be good to be able to have my sample, but run it later. And we have a kit coming out next year in the form of, we call it a fixed RNA kit. So it's basically where you're going to be able to generate a suspension of single cells. So you still need to do the sample prep, but then you can fix it. And once it's fixed, it'll be stable for a long period of time, so that you can kind of either batch analyze your samples, or do that sample prep late at night, and then not have to worry about getting straight to the Chromium. You can go and store your sample to basically when you're ready to run it. So we've definitely got some exciting details upcoming about that assay. It will be a gene expression based assay that allows you to measure cell surface protein as well, and ultimately it'll be probe based though, rather than capturing the transcripts directly, because the fixation processes itself can damage mRNA molecules. So in our other products, we capture the mRNA molecule directly. Here it'll be a probe based product, where we're going to have a probe that binds to a transcript if it's present, or maybe washed away if it's not present, and then we'll capture that probe through this fixed RNA solution. So we do have kits coming within this area to help with some of these issues.
Yeah, that will help a lot, because our experience is that you plan 1 hour of experiment and it always is the double.
Yeah. Oh dear, I think we lost Cátia. Next question, are there major differences if we look at the transcript between those from nuclei versus whole cell? It's an interesting question, one that gets asked quite often. We find, well, not we, the research community has generally found that transcripts that are captured from nuclei do represent what is present in whole cell, but they are slightly different in that you get a lot of nascent transcripts, so unspliced transcripts, because you're in the nucleus now as opposed to the cytoplasm. So you do see a lot of higher intron retention within those transcripts, but in general, the signal that you observe from nuclei is reflective of the whole cell. I don't know if Uyen or Cátia, you have any experience here examining differences between signal obtained from nuclei versus signal obtained from whole cells?
I was a little bit lost, frozen without internet.
Yeah, so so the question is, someone asked about the major difference between the transcript between the nuclei and the whole cells.
Yeah, so there is a little bit of loss when because you are losing the cytoplasm. But normally, with all the data that there are already with total cells, computationally, you can really like input is the word, the data. But yeah, again, ideally, my opinion is like if you can get cells, total cells, yeah, that is ideal. If not, yeah. For example, again, for brain cancers, it's very difficult to get total cells. And even if you isolate total cells, then the data sometimes are not really what you expect.
even if you see that the cells are viable, sometimes, yeah, they are a little bit dying already. So, yeah.
Next question is, isolating live cells from low viability samples, would/could this compromise data to bias against cell populations that are more susceptible/more fragile?
Yes, yeah. And yeah, I agree, it will. But the thing is that what I mentioned, if you have more sample, for example, if you are working with a type of tissue that you can get other sample preparation, just try to optimize and to get more like the high viable cells. If it's a sample from a patient and you don't have any other option, you just have cells that the viability is low, yeah, you need to do it. You don't have other option. So it's a little bit always, depending what the conditions that you have, the sample that you have, you need to do these quick decisions on all the experiments that you do. And for me, optimization is the key. Try to optimize with samples, a mock sample that you don't need to for the experiment, but yeah, at least until you get viable cells.
Yeah, that's definitely excellent advice. If
Yeah.
if you're fortunate enough to have samples where you've got a lot of material or you can access it very readily, even if it's not the same sample over time, performing some upfront optimization of that sample prep is really critical. Ending up with a nice, clean, viable single cell suspension is key to a successful experiment. Obviously, there's cases where that can't happen, patient-derived samples, rare samples, but understanding your sample prior to a single cell experiment is really going to set you up for success here.
Yes, and even if at the end you cannot get enough cells or enough, it's like try to search for other solution. Single cell, it's good, it should be done, but sometimes it's not possible. And in my opinion, it's like you need to be conscious of the sample that you have. If it's not possible, don't do it, because you will spend time, money, and at the end, no one will be able to save the data that you have. So yeah.
Great. Well, honestly, this has been a great discussion. We're running over time, so there's no extra questions. Happy for everyone to get back to their busy day. But thank you so much, Cátia and Nguyen, for presenting today.
Thank you.
And looking forward to the next session, which I believe at this stage we're planning to give more of a general overview about the single cell gene expression and single cell immune profiling products and what their capabilities are, as well as what our new products we have coming in the pipeline, and hopefully also hearing from perhaps a customer who's use these products in their experience in their hands.
Thank you, Geoff.
Thank you so much. Thank you all.
Take care, everyone.
Bye. See you. Thanks.